You know, for the longest time, we all thought of RNA as just DNA's simple delivery boy. A temporary messenger, that's it. But it turns out that's not even half the story. RNA is a secret agent, a master editor, and a powerful controller of our entire genetic code. So let's dive in and uncover this hidden world, one incredible secret at a time.
Our story starts with something you probably wouldn't expect: a petunia. See, scientists wanted to make these flowers an even deeper, richer purple. So they added extra copies of the gene for purple pigment. Logically, more gene equals more purple, right? But instead, they got this: patches of pure white. They added the instructions for purple and somehow ended up erasing it.
And that right there is our central mystery. How is that even possible? How can you put more genetic information in and get less of the result out? I mean, this paradox just baffled researchers, but the answer was about to completely rewrite our understanding of how genes are controlled.
Yeah, that petunia puzzle was no fluke. It was the very first clue that our cells have this powerful built-in security system, a way to silence genes with stunning precision. And the key player, the one running the whole operation, wasn't DNA. It was its often overlooked cousin, RNA. This whole system is called RNA interference, or RNAi for short. And you can think of it like a molecular search and destroy mission. The cell cleverly uses the sequence of a genetic message against itself, hunting it down and shredding it before it ever gets the chance to be turned into a protein. So how does this whole thing work? Well, it kicks off when a double-stranded RNA molecule shows up. To a cell, that's a huge red flag. It often means a virus is invading. Immediately, an enzyme called dicer jumps into action, acting like a paper shredder, chopping that threat into tiny little pieces. Then these pieces get loaded into a protein machine called RISC. And here's the brilliant part. RISC uses that little RNA fragment as a GPS, turning the whole complex into a heat-seeking missile that hunts down any matching messenger RNA and, snip, message destroyed, problem solved.
And that brings us right back to our petunias. By cramming in so many extra copies of that purple gene, the scientists accidentally made the cell produce double-stranded RNA. The cell's security system kicked into high gear, saw a threat, and unleashed the RNAi pathway, destroying the purple message entirely. So, mystery solved. But believe it or not, silencing is just one of RNA's secret jobs. So RNA can destroy a message. That's pretty amazing. But what if it could do more? What if, instead of just deleting a message, it could fundamentally change it after it's already been written? We're talking about an editor that doesn't just cross out sentences, but actually rewrites them with brand new words.
And as Dr. Sat Saywert points out, this isn't just a minor tweak. RNA editing shows us that the road from a gene to a protein is way more dynamic and honestly more creative than we ever imagined. The cell isn't just blindly following instructions, it's improvising. And some of these edits are just wild. I mean, in certain parasites, the messenger RNA starts out as practically gibberish. The cell's machinery has to go in and stitch in dozens of new genetic letters just to make it readable. And get this, in our own bodies, special enzymes perform a kind of molecular surgery, changing one genetic letter into another. One tiny swap can completely alter what the final protein does.
And make no mistake, this is a life or death process. In your brain right now, this exact type of editing is happening. An enzyme is making a single letter change in the blueprint for a vital brain receptor. Without that one tiny edit, the channel it builds goes haywire, it lets way too much calcium into your neurons, and that is fatal. Our lives literally depend on RNA's editing skills. Just imagine what understanding this could mean for future therapy.
Okay, so beyond editing single letters, RNA also acts as a master assembler. It takes these huge, raw genetic transcripts and carves them up with incredible precision. It's like a sculptor looking at a big block of marble and knowing exactly where to chip away to reveal the masterpiece inside.
Take the RNA that builds our protein factories, the ribosomes. It all starts as one long, unwieldy piece of RNA. Then the cell goes to work, making a series of precise cuts, a snip here, a snip there, whittling it down into smaller and smaller pieces. This keeps going until finally 3 distinct, perfect, mature RNA molecules pop out, ready to be assembled. It's just a beautiful example of cellular efficiency. Now, we might be familiar with the standard way of doing things, called cis-splicing. That's where you stitch together different pieces from the same gene transcript. But some organisms do something that sounds like it's from a sci-fi movie: trans-splicing. This is where the cell takes a piece of RNA from one gene over here and stitches it onto a piece of RNA from a completely different gene over there. It's like taking a page from one book and pasting it into another to create a totally new story. It's just incredible.
So, RNA can be silenced, edited, and reassembled. But what about timing? Well, it turns out, one of the most powerful ways to control a gene is to simply decide how long its messenger RNA is allowed to stick around.
Let's think about a real-world problem your body solves every second. Iron. Too much is toxic, but too little is really dangerous. Your body needs to perfectly control the protein that brings iron into your cells. So how does it do it? The secret is all about mRNA stability.
And the control system is just so elegantly simple. Check it out. When your iron levels are low, a special protein binds to the messenger RNA and acts like a shield, making it super stable. Since that message sticks around for a long time, your cell can make lots and lots of iron-importing proteins. But when your iron levels get high, the iron itself forces that protein to let go. Now the mRNA is unprotected and gets destroyed really quickly. Less message means less protein. It's a perfect feedback loop. This whole regulatory dance happens at these special little landing pads on the messenger RNA itself, called iron response elements. These are basically little hairpin loops of RNA that act as the control switch, the place where that protein binds. It's what allows the cell to fine-tune protein production in real time based on exactly what your body needs at that moment.
So when we step back and look at everything, we've seen RNA act as a silencer, an editor, an assembler, and a timer. It's really clear that we need to update our thinking. RNA isn't just a simple messenger. It's a dynamic, powerful regulator at the very heart of how our genes work.
Probably the best way to think about it is like this. DNA is the master blueprint, locked away safely in the architect's office. It's stable, protected, unchanging. But RNA? RNA is the working copy you take out to the construction site. It can be marked up, edited, changed, or just thrown away when you're done. It's this amazing flexibility that allows for the incredible complexity of life.
So in the end, we've learned that RNA wears a lot of different hats. It's a gene silencer, wiping out specific messages. It's a code editor, rewriting genetic instructions on the fly. It's a master assembler, stitching together totally new creations. And it's a production controller, managing how much of a protein gets made just by controlling its own lifespan. RNA is running a huge part of the show.
This total revolution in how we see RNA has opened up a universe of new questions. It is so clear that this molecule is way more central and powerful than we ever knew. And it just leaves you wondering, if we've uncovered all of this in just the last few decades, what other incredible secrets are still locked away in the secret life of RNA?